beauty of bacteria and all living things. We all use A, G, C, and T. It doesn't matter. Our structure of our DNA is identical across the planet. All right, so we can manipulate DNA and use it for our purposes because we can insert it into different organisms, creating transgenic organisms. Okay, so in this case with the bacteria, okay, we have our plasmid. We have little enzymes that have the ability to cut DNA at certain points, certain base pair routes. All right, usually we use one called EcoR1. EcoR1 cuts and looks for a pattern of GGACT. When it finds that pattern, it sticks to the DNA and cuts it right there, all right, and then opens it up. And so that's what we're doing with the plasma of the bacteria. We've isolated the gene of interest that we want to insert into that bacteria, and all we do is mix the bacteria and the gene together, and the gene will assimilate into the bacteria through a process of transformation, all right? Bacteria do this. They suck up random DNA all the time. They don't care. Okay, and in that process, we're going to do that. We're just going to trick the bacteria into doing it. Right? We're going to heat shock them. We're going to cold shock them. And when we do that, it opens up their membrane and allows that DNA that we want to get right into the bacteria. It's not perfect. Our transformation rates are low. They're usually 10-15%. Okay, but we still get some of that gene in there. Okay, so once the gene gets in there, we create a recombinant DNA plasmid. All right, which is in that bacterium. And then the bacterium, we grow that bacterium, or we go through asexual reproduction of that bacterium in a culture, okay? So wherever that culture is, we do that. It grows and grows and grows and grows. Then there's two things that we do, okay? We take the proteins that DNA created, right, which we're going to talk about next chapter, and we're going to take that protein and we're going to use those proteins for various things. For example, people that have growth issues, have pituitary issues, that cause them not to grow well, and they have a shortage of human growth hormone, HGH. We actually insert the gene for HGH production in a bacteria. The bacteria then turns out HGH protein. We harvest that protein and we use it to help kids that have pituitary issues. Or a protein that dissolves blood clots and heart attack therapy. Same thing, we know where that gene is, we put that gene in the bacteria, and the bacteria in the culture just churns out that protein, and we use it for that purpose. Or we actually use the gene, all right, and we alter the bacteria, and in this case, we're altering the bacteria because we've created bacteria that eat up oil, all right, so they've been known to be put on oil spills and they dissolve and take care of oil, all right, or resistance into plants, all right, pesticide resistance into plants, or for example, the indigo dye that you see in blue jeans comes from bacteria. Indigo dye is from a plant. We don't have enough indigo plants produce enough dye. And so we've taken that gene out of the plant, inserted in the bacteria, and now they have these huge vats of bacteria, and they just churn out indigo dye using that indigo dye protein. Okay? So that's what we're talking about with gene cloning. Not animal cloning, that's a whole different realm. Okay, it's a whole different thing. But this is a big application that we're currently using frequently, right, to work on and isolate gene piece. And this isn't even, when we get through 15, we're going to do a little thing on CRISPR, okay? And if you've read anything about CRISPR, it's crazy and it's scary.